So today we're going to be talking about Chapter 3, Neurophysiology. For starters, what is neurophysiology? Neurophysiology is a study of electrical and chemical process in the neurons. So to get started, we should talk about ions, anions, cations, intracellular fluid, and we'll keep it going. Ions are electrically charged molecules. Anions are negatively charged ions. Cations are positively charged ions. Intracellular fluid dissolves these ions and spreads it around in the middle. It's separated from extracellular fluid through a cell membrane, basically a lipid bilayer. So scientists up here used something called a microelectrode to test whether the inside of the neuron was negatively charged or positively charged, just to get a feel for it. So what they ended up figuring out was the inside of the axon has a more negative charge than the outside axon when it's resting, like at a resting potential. So you might be thinking, what is this resting potential? It's basically from negative 50 millivolts to about negative 80 millivolts. What else? We have something called ionic channels. There are proteins that allow specific ions through membranes to and from. It only selects, for example, in this one, these yellow particles will go through. These big blue ones aren't allowed through. Selective permeability, basically. So, neurons have leaking channels that always allow sodium and potassium and other materials in and out of the membrane at all times. Just keep that in the back of your head. So when it comes to permeability and stuff like this, we have to talk about diffusion. So diffusion is material moving from a higher concentration to a lower concentration. Alright, so now we need to talk about a concentration gradient. Where's that out? Here we go, concentration gradients. Concentration gradients, you should picture it like this. Think of rolling a rock up a hill. That would take energy to push it up, right? Now think of pushing a rock down a hill, but it takes almost no energy to do so. It's basically the flow of It's the flow of concentration of a substance in a region that can flow up or down and balance itself out. This right here, the concentration gradient, okay, well, from my eyes, it looks like this side has a lot more yellow particles than this side. So, the concentration gradient might move down to give this side an equal amount of particles as well. Now, if we're going to talk about the other way that concentration gradients work, you can move up a concentration gradient. And as I said before, moving up is like pushing a rock up a hill. It takes energy, some ATP. For example, if we wanted to get these blue atoms or ions over to the other side, we'd have to put energy into it to move it up the gradient. Let's find sodium-potassium pumps. Here we go, sodium-potassium pumps. Basically deal with concentration gradients too. So this pump pushes the Na out and pumps K plus ions in, potassium ions. So pumps in 2 potassium ions and pumps out 3 sodium ions. These Na molecules aren't let back into the cell. Only K plus has the ability to go back and forth in and outside of the cell. And basically this is why this reaction right here, these proteins, these ion pumps, is what creates a resting membrane potential. As I said before, it's between negative 50 and negative 80 millivolts. So, what's the actual representation of this resting membrane potential? So basically, sodium leaves the cell. It pumps in 2K plus ions, however. So The cell membrane has an ionic channel to allow K plus to go in and out of the cell and up and down its concentration gradient. Remember, going up requires energy. Going down really requires close to nothing. So when K plus leaves the cell and goes out, it gives the inside of the neuron a less positive charge. And when you keep on taking away positive charges, you, in a way, make the inside more negative because you've taken away a positive charge. So when enough K plus leaves, it brings a membrane potential to about negative 60 millivolts, and K plus has a balanced out gradient. And it kind of would like stop this from occurring. It would stop K from passing through membrane. There's not really a good picture for it here, but I can always keep on explaining it. So picture this. You have equal amounts of K on both sides, and they keep on going back and forth, correct? So the K would keep on going in and out of the membrane, making the inside more negative. Once the inside gets to a certain point of negativity, it's going to start repelling the income of the potassium ions. It'll actually stop diffusion from happening. The concentration gradient will be balanced out. You don't want anything happening. So, Nernst equation is a mathematical equation for predicting when this can actually happen with these K plus ions. Now here's this huge equation for it. And it turns out it happens at negative 80 millivolts for the equilibrium potential. Stop letting the influx of K plus ions in. With this Nernst equation, see how it only takes for account the K plus ion. The ion of interest in here is the K plus. But for the Goldman equation, which is not shown here, it takes into account both sodium and potassium ions. But it basically does the same thing. Another important thing we have to talk about, calcium ions. So, calcium ions are usually kept inside the neuron for intracellular signaling, basically between synapses and all that, knowing when to release neurotransmitters and when not to. Quick backtrack. A good thing to remember is the amount of concentrations of ions in and outside of the cell. Sodium really is outside of the cell. It's really highly concentrated out there. Not so much for potassium or chlorine or that either. It's basically the opposite. So just remember that. Keep that in mind. What next? Remember, here's a quick side note. All cells have a resting membrane potential. It's just neurons are the ones that can choose to change those potentials. All right. Now we're going to go through a threshold amount of depolarization, triggers, and action potential. All right. So, action potentials. What are they? They're brief neuronal polarization shot down from an axon hillock to send messages to other neurons. All right. So how did they do that? They go through hyperpolarization and depolarization. So hyperpolarization increases negativity. It makes it go farther from zero, negatively. Depolarization decreases negativity. It moves closer to zero, positively. Think polarization equaling negativity, and hyper equals more, and D equals less. All right, let's move on to local potentials. Okay. Local potentials. Think about dropping a pebble into a peaceful pond and seeing ripples flowing throughout that whole entire pond. These ripples spread far and wide, but eventually die out. The same thing happens in an action potential wave. It slowly dies out. All right, next next we're going to have to talk about thresholds. Now, what is a threshold? Threshold is basically in a neuron. When a neuron is depolarized to a point of negative 40 millivolts. Let's see an example of a threshold. Okay. Here we have the threshold of a neuron, correct? Negative 40 around that area. So right now it's hyperpolarized. Once you get higher and higher and reach this point, shoot up for an action potential, and you have a refractory period. Alright. Larger depolarizations create more action potentials. Not larger action potentials, just frequency. And information is transferred through the frequencies. So, yeah. So, let's go over. Psych. We have an all or nothing property. An action potential either fires or doesn't fire. It either has enough substrates to make it go past threshold or not. Now, after potential, as we see here, is the cannot talk today. The hyperpolarization right after the spike of an action potential is basically a refractory period. Everything is like, nothing is coming in, nothing is coming out. Not always true though, because as I said before, remember, we always have leaking protein channels and ion channels that allow sodium, potassium, and other various materials in. So after a certain period of time, you see how this curves down and works its way up. Those leaking channels are working their way to bring the neuron back to what it once was. And then the process starts over again. An example from what I said before about the strength of depolarizations. This is what happens when you get a strong depolarization. You get a large frequency. If you get something that gets you just above threshold, you only get like one frequency. But if you put in more power into it, you get a whole bunch more. These frequencies are what communicates with other neurons. And that's all, folks.